Dear friends, good morning to all. Greetings from Dr. G. Sivayalangu. Today we can discuss about antigen antibody reaction. Introduction. Antigens and antibodies combine with each other specifically and in an observable manner. In the laboratory, they help in the diagnosis of infections, in the identification of infectious antigen and also non-infectious antigen such as enzymes. The reactions between antigen and antibodies occur in three stages. They are primary stage, secondary stage, tertiary stage. Primary stage means it is the initial interaction between the antigen and antibody without any visible effects. Secondary stage means leads to demonstrable events such as precipitation, agglutination, lysis of cells, killing of line antigens, neutralization of toxins. These are secondary stage. Next one, tertiary stage. Tertiary stage leads to neutralization or destruction of infectious antigen and humoral immunity against infectious disease. Next one, properties of antigen antibody reaction. The reaction is specific one. The entire molecule react and not a fragment. There is no denaturation of antigen or antibody during the reaction. The combination occurs at the surface of molecule. The combination is film but reversible one. Both antigen and antibodies precipitate or agglutinate, resulting in the formation of agglutinates or precipitates. First we will see about precipitation. When an antigens, soluble form of antigens reacts with antibody in aqueous solution, which forms a latex that eventually develop into precipitate. This reaction is called as precipitation. Next, precipitin. Antibodies that aggregate soluble antigens are called as precipitin. Next, move on to precipitation curve. This plot of the amount of the antibody precipitated versus increasing antigen concentrations at a constant total antibody, which reveals three zones: a zone of antibody excess and an equivalence zone. Third one is a zone of antigen excess. Under these conditions, antibody excess or antigen excess extensive lattice do not form and the precipitation is inhibited. Whenever antigen or antibody concentration is excess means that time no inhibitions there, precipitation is there. Although the quantitative precipitation reaction is seldom, rarely used experimentally today, the principles of antigen excess, antibody excess and equivalence apply to many antigen antibody reactions. Next, conditions for precipitation. Antibody must be bivalent one. The antigen must be either bivalent or polyvalent. Next, precipitation curve. The precipitation reaction can be quantitatively measured by precipitation curve. It can be used to measure the amount of antigen or antibody present in the sample. This is the picture shows the precipitation curve. First one, antibodies excess zone. In between, zone of equivalence. Zone of equivalence is antibody antigen concentration equal 1. Third one is antigen excess. Next, the precipitation steps. A constant amount of antibodies are added to series of test tubes. To it added increasing concentration of antigen. Precipitation forms after incubation. Thus, the tubes were centrifuged. After discarding the supernatant, the amount of precipitate is measured. The maximum precipitation occurs at the zone of equivalence, within which the ratio of antibody antigen is optimum. Optimum in the sense antigen antibody concentration are equal one. When antibody or antigen is excess, lattice do not form and precipitation is inhibited. This inhibition can be seen in zone of antibody excess and the zone of antigen excess. Next, move on to agglutination. The interaction between antibody and a particulate antigen resulting in visible clump formation are called as agglutination. Next one, agglutinin. The antibodies which produce agglutination are called as agglutinin. Next, prozone effect. The apparent decrease in an antigen antibody reaction sometimes observed as the concentration of antigen or antibody is increased. Just as an excess of antibody which inhibits the precipitation reactions, such excess can also inhibit the agglutinations. This inhibition is called as prozone effect. Next, examples of agglutination. Heme agglutination. Agglutination reactions are very much useful in blood group typing. The human RBC contains antigen A and B. When these RBC cells mixed with antisera A or B, they result in agglutination, forming visible clump on the slide. This determination of antigen on donor and recipient RBCs helps during the blood transfusion time. This picture shows the blood grouping. Here this slide containing A, B, RH. First line shows the A plus means A positive. Here agglutination occur A and RH only. So this is for A positive. Second one, the agglutination occur in B and also RH. So this B positive. Third one is no agglutination occur in A and B only for RH. So O positive. Fourth one is agglutination occur in A, B as well as RH also. So this is called as AB positive. In this way we are find outing the blood grouping. Next one, bacterial agglutination. Diagnosis of bacterial infection. Example for Vidal test for diagnosing Salmonella typhi infection. During bacterial infection, antibodies are produced against major surface antigen of organism. These antibodies are detected by bacterial agglutination. The patient's serum sample, which contains antibody, is added with bacteria containing surface antigen. The positive sample, which shows the agglutination. Next, the pictures shows the Vidal test. Here the slide containing 6 circles. First one O antigen, second one H antigen, third one AH antigen, fourth one BH antigen, fifth one NC means negative control, PC means positive control. In the sample containing O or H or AH or BH, compared to positive, the agglutination like positive control means the particular antigens are present. In this way we are finding out the Vidal Salmonella typhi antigens. Thank you friends. Thank you for your support.